Welcome to A-Minder, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello, I'm Elyn, and I'll be your host for this episode. This is an episode from the Disease Mechanisms of Alzheimer's series for June 2020, and it's split into two segments. The first segment is all the papers about cerebrovascular changes relating to Alzheimer's disease, and the second segment is called Unclassified Disease Mechanisms, where I'll cover all of the other disease mechanisms papers that didn't quite slot into our other categories. While it's a pretty mixed bag of papers, I've done my best to sort them into meaningful categories along the way. So since my PhD thesis is largely focused on cerebrovascular changes in AD, I'm going to start with that segment, which includes 7 papers from the month of June. Beginning with papers related to cerebral amyloid angiopathy, or CAA, which is the deposition of amyloid into cerebral vessels, we have a paper titled Amyloid Beta Plaques May Be Reduced in Advanced Stages of Cerebral Amyloid Angiopathy in the Elderly. This is a paper by first author Okamoto and last author Takatama, published in Neuropathology. This paper was a case study of 29 CAA cases in Japan, in which 2 had notable characteristics. These 2 cases had few parenchymal amyloid beta, or A-beta, plaques, and extensive CAA in small vessels and capillaries throughout the brain, with the exception of the frontal lobes. Immunohistochemistry staining for GFAP and A-beta42 revealed that some astrocytes appeared to contain A-beta, and staining for IBA1 revealed the presence of microglia around some small vessels with CAA, which also seemed to have phagocytosed A-beta. Some of the remaining plaques seemed to have a moth-eaten appearance, and the accumulation of T lymphocytes was also observed in the subarachnoid space in one of the cases. Overall, the morphological changes observed in these 2 cases appeared to be similar to those post-amyloid immunotherapy, although neither case was treated with this therapy. This suggests an innate mechanism of amyloid clearance in advanced CAA cases. This will be important to understand moving forward, especially as Biogen's amyloid immunotherapy aducanumab is being filed for FDA approval, so a very interesting study. The next paper is a very exciting one, published by first author Blanchard and last author Tsai in Nature Medicine, which is titled Reconstruction of the Human Blood-Brain Barrier in Vitro Reveals a Pathogenic Mechanism of ApoE4 in Parasites. So there's a lot to unpack here, as the group both generated a novel in vitro model of CAA and made significant progress in uncovering why ApoE4 is the strongest genetic risk factor for capillary CAA. Their novel model, which they call the IBBB, is an induced pluripotent stem cell-derived system comprised of endothelial cells, pericyte-like mural cells, and astrocytes, which self-assemble in a 3D matrix to model brain microvessels. When the cells comprising the IBBB were of ApoE4 genotype, they found significantly more amyloid accumulation around the in vitro vessels. They also found that this seemed to be specific to the inclusion of ApoE4 mural cells, or pericytes, in the model, and wasn't affected by ApoE4 genotype in the other cell types. So they delved into this further and found that dysregulation of the calcineurin and nuclear factor of activated T cells, or NFAT, signaling in ApoE4 pericyte-like mural cells increased the ApoE expression of those cells. And this increase in ApoE protein seemed to drive the increase in amyloid accumulation. They validated these in vitro findings in human brain samples and showed that inhibiting the calcineurin and fat signaling pathway reduced amyloid accumulation in their model. This was a really interesting paper with lots more to unpack than is mentioned here, and it's definitely worth a read if you're in this field. I really enjoyed it and actually presented it at one of my lab meetings for a journal club. So straying from CAA but keeping in line with mural cells, next we have a paper titled Excessive Production of Transforming Growth Factor Beta-1 Causes Mural Cell Depletion from Cerebral Small Vessels. This one's by first author Kato and last author Onodera, published in Frontiers in Aging Neuroscience. So cerebrovascular health heavily relies on intact mural cells, since they have many critical functions including regulating cerebral blood flow and the blood-brain barrier. Dysregulated TGF-beta, or transforming growth factor beta, signaling has previously been connected to cerebrovascular pathologies. But this group set out to determine whether increased TGF-beta production resulted in the degeneration of vascular mural cells. They used a mouse model overexpressing TGF-beta1 under the GFAP promoter and found that chronic overproduction of this protein resulted in a reduction of both smooth muscle cells and pericytes covering the vessels. This degeneration was also accompanied by vasodilation. So this provides important insight into mural cell degeneration in pathological conditions, which is currently a very topical topic in the field. Moving into some cerebrovascular changes found with MRI, we have a paper called Restricted Effect of Cerebral Microbleeds on Regional Magnetic Susceptibility. This is by first author Fazlohi and last author Eyton, published in Journal of Alzheimer's Disease. So cortical iron accumulation has been reported as a pathological feature of Alzheimer's disease, but the cause of this remains largely unknown. This study aimed to figure out whether hemosiderin deposits from cerebral microbleeds can contribute to this accumulation. The group used 32 MRI scans found to have cerebral microbleeds and used an MRI technique called quantitative susceptibility mapping to estimate the iron content in the tissue surrounding the microbleed. Overall, they found that the susceptibility change around the microbleed was too small of a region to account for the regional iron changes that are routinely seen in the cortex of patients with AD. So it may not be the cerebral microbleeds that are driving the iron accumulation. Sticking with MRI, we have a paper called White Matter Lesions and Temporal Atrophy are associated with cognitive and neuropsychiatric symptoms in patients with hypertension and Alzheimer's disease. This one's by first author Garcia Alberca and last author Garcia Caceres, published in the International Journal of Geriatric Psychiatry. In an attempt to further explain the relationship between AD and hypertension, this group studied 92 patients with AD and looked for associations between hypertension, white matter hyperintensities, and medial temporal atrophy, among other variables. They found that AD patients with hypertension had significantly worse cognitive status and significantly more white matter hyperintensities and medial temporal atrophy than AD patients without hypertension. There was also one paper on molecular changes with implications in the cerebrovasculature, which was titled Alzheimer's Amyloid Beta and Tau Protein Accumulation is Associated with Decreased Expression of the LDL Receptor Associated Protein in Human Brain Tissue. This one was by first author Shepard and last author Halliday, published in Brain and Behavior. So the LDL receptor associated protein, also known as RAP, has previously been shown to bind A-beta and enhance its clearance through cellular uptake. This study aimed to determine whether RAP levels are altered in AD human brain tissue and whether they are related to the level of A-beta and tau in the brain. To do this, the authors quantified RAP, nu-N, LRP1, tau, and A-beta levels in the temporal cortex of 17 AD and 16 control cases. They found that RAP levels were significantly decreased in AD brain tissue, and that levels of RAP were significantly negatively correlated with A-beta and tau pathology. nu-N and LRP1 levels didn't show any correlations. So this suggests that accumulation and aggregation of A-beta may, in part, be due to reduced RAP levels. To finish off this segment on cerebrovascular changes, we have a paper on a new method called Quantitative Assessment of Age-Associated Alterations in Brain Vasculature in Wild-type mice and in Bigenic mice that model Alzheimer's disease. This one was by first author Govertz and last author Himmelreich, published in Molecular Imaging and Biology. The magnetic resonance angiography, or MRA, is one of the most common methods used to investigate vascular dysfunction in mouse models. But the processing of this type of data has not yet been streamlined. This paper describes a semi-automated processing pipeline that can improve the classic labor-intensive and observer-dependent methods that are classically used to deal with MRA data. In their study, they demonstrated that subtle changes in vessel morphology captured on MRA images from an animal MRI scanner can be non-invasively quantified with this pipeline. That's all for the new papers related to cerebrovascular changes in Alzheimer's, and I hope you enjoyed this segment. For the month of June, we've sorted 20 papers into unclassified disease mechanisms. As I've said, it's quite a mixed bag, as you'll see, but I've done my best to sort them into meaningful chunks. To start this section, I've got a few papers related to RNA. The first one is titled, Alzheimer's Disease-Related Dysregulation of mRNA Translation Causes Key Pathological Features with Aging. This one's by first author Ghosh and last author Gies, published in Translational Psychiatry. As alluded to in the title, this study investigated whether changes in signaling related to mRNA translation can lead to AD-related pathological features. In an initial screen, the group found that treating primary neurons with A-beta in the nanomolar range led to increased FMRP-regulated protein synthesis. They determined that the selective regulation of mRNA translation reduced CYFIP2, or CYFIP2, as I'll call it, which is an FMRP binding protein. When they modeled the CYFIP2 reduction in mice, they observed age-dependent key pathological features of AD. With this, they postulated that prolonged activation of this pathway may be an early step in the progression to AD, and that this pathway may be a viable therapeutic target. The next paper is focused on microRNA, and it's titled Induction of the Endoplasmic Reticulum Stress Response, microRNA 34A Targeting of the IRE1 Alpha Branch. This one's by first author Krems and last author Mies, published in Cells. Focused on the endoplasmic reticulum unfolded protein response as a pathway that may exacerbate AD, this group demonstrated that a specific miRNA, which was MIR34A5P, which is upregulated in the brains of patients with Alzheimer's disease, can regulate this unfolded protein response. When the group induced endoplasmic reticulum stress in neuronal cells, they found that MIR34A5P overexpression significantly reduced the IRE1 alpha and XBP1S, which led to decreased viability and increased caspase activity. They proposed that it's possible to target the entire IRE1 alpha branch of the unfolded protein response through modulation of this single microRNA. Next, we have one on long non-coding RNA, titled Knockdown of Long Non-Coding RNA TUG1 Depresses Apoptosis of Hippocampal Neurons in Alzheimer's Disease by Elevating microRNA 15A and Repressing ROC1 Expression. This was by first author Li and last author Kong, published in Inflammation Research. This study aimed to pinpoint how the long non-coding RNA taurine upregulated gene 1, or TUG1, can affect hippocampal neurons in the context of Alzheimer's disease. Specifically, they investigated how TUG1 can modulate MIR15A and Rho-associated protein kinase 1, or ROC1, in a combination of mouse and cell culture models. In their AD mouse model, they found that both TUG1 silencing and increases in MIR15A improved cognition and underlying pathology, specifically strengthening antioxidant ability of hippocampal neurons. In their cell culture model, they found that TUG1 silencing and increases of MIR15A increased viability and limited apoptosis. They also observed that TUG1 specifically bound to MIR15A, and MIR15A targeted ROC1. Overall, they concluded that TUG1 reduction restricts apoptosis in A-beta 25 to 35 treated hippocampal neurons by increasing MIR15A and suppressing ROC1 expression. So completing the RNA tour, the next one is on circular RNA, and it's titled Circular RNAs Regulate its Parental Genes Transcription in the AD mouse model using two methods of library construction. This one's by first author Ma and last author Wan, published in FASEB Journal. To expand the wealth of knowledge relating to the participation of circular RNAs in Alzheimer's disease, this group profiled brain circular RNAs in wild-type and APP-PS1 mice. They used two types of samples to detect the circular RNAs in the brain tissue. The first one was ribosomal RNA-depleted samples, which I'll call the rRNA negative library, and the second one was ribosomal RNA-depleted samples that were also treated with RNase R to digest any linear RNA. This one's called the RNase positive library. So they detected more circular RNAs in the RNase positive library and found that it more clearly revealed differentially expressed circular RNAs compared to the rRNA negative library. They found significantly dysregulated circular RNAs between the wild-type and APP-PS1 mice, and a KEGG analysis showed that the dysregulated circular RNAs may be involved in AD from many different angles, including cAMP signaling, axon guidance, and long-term potentiation, among others. They noted two specific circular RNAs that regulated the parental genes TRPC6 and NME7 respectively. Next, we have a couple of methods, the first one being an enzyme-free amplification strategy based on 2 photon fluorescent carbon dots for monitoring MIR9 in live neurons and brain tissues of Alzheimer's disease mice. This is by first author Wu and last author Tian, published in Chemical Communications. This paper presents a novel method for monitoring the miRNA MIR9 in live neurons and brain tissue of AD mouse models. The method leverages an enzyme-free amplification strategy based upon 2 photon fluorescent carbon dots. Using their new method, they found that MIR9 was upregulated in 6-month-old APP-PS1 mice and downregulated to lower than wild-type mice in 15-month-old APP-PS1 mice. The next paper is another method paper, which is called Object-Based Analyses in Fiji/ImageJ to measure local RNA translation sites in neurites in response to A-beta 42 oligomers. This is by first author Gamera and last author Ballariola, published in Frontiers in Neuroscience. This group presents a novel method to visualize and quantify local translation events in polarized cells with many extensions, like neurons. The method is an object-based analysis where newly synthesized proteins are tagged with puromycin, and the SYTO nucleic acid stain is used to label endogenous RNA. The paper describes the algorithm used to identify co-localization of newly synthesized proteins and RNA in cells. They validated their novel method by comparing the local translation events in control neurons compared to A-beta 42 treated neurons. They found that A-beta treatment increased neuritic protein synthesis and the fraction of translated RNAs in distal sites of the neurite, suggesting an induction of local protein synthesis. Sticking with RNA but focused more on RNA binding proteins now, we have a paper called RBM45 associates with nuclear stress bodies and forms nuclear inclusions during chronic cellular stress and in neurodegenerative diseases. This is by first author Collins and last author Bowser, published in Acta Neuropathologica Communications. This group set out to further investigate nuclear and cytoplasmic inclusions of the RNA binding protein RBM45, which are present across many neurodegenerative diseases, including AD. Specifically, they investigated whether the protein functions via association with membraneless organelles in cells, and whether these associations could promote the formation of inclusions. They found that under normal conditions there were no evident inclusions, but under cellular stress, RBM45 reversibly incorporated into nuclear stress bodies, which was dependent on its binding to RNA. They also observed that under chronic stress, RBM45 irreversibly accumulated in nuclear inclusions. The group then quantified cell type and disease-specific patterns in RBM45 pathology and found inclusions in both neurons and glia in AD samples that were not present in controls. Next on to another paper on RNA binding proteins, which is titled SFPQ and Tau, Critical Factors Contributing to Rapid Progression of Alzheimer's Disease. This one's by first author Eunice and last author Zaire, published in Acta Neuropathologica. In this study, the authors used a combination of RNA pull-down and proteomics to identify disease subtype-specific alterations in the RNA binding proteins. They assessed the RNA binding proteome of sporadic AD, rapidly progressive AD, and sporadic Creutzfeldt-Jakob disease alongside control cases. They found that one RNA binding protein called splicing factor proline and glutamine rich, or SFPQ, was downregulated in rapidly progressive AD patients, Creutzfeldt-Jakob disease patients, and the 3 times transgenic mouse model of AD at the terminal stage of the disease. In contrast, at an early disease stage in the 3 times TG mouse model, SFPQ was upregulated. Interestingly, the group found that in the brains of rapidly progressive AD patients, the protein was dislocated from the nucleus and was actually co-localized in the cytoplasm with phospho-tau. They followed this observation with an in vitro study which showed that SFPQ is recruited into TIA1 positive stress granules after oxidative stress induction, where it co-localizes with tau and p-tau. They also found that tau expression was able to reduce SFPQ expression, which may explain the downregulation of the protein they observed in late-stage postmortem samples. Overall dysregulation and dislocation of SFPQ and its association with pathological tau seems to be a critical pathway contributing to the rapid progression of AD. The next paper is titled Heme Oxygenase 1 Inhibits Neural Stem Cell Proliferation and Survival via Modulation of the WNT beta-catenin signaling. This is by first author C and last author Gao, published in Journal of Alzheimer's Disease. So heme oxygenase 1, or HO1, is a stress-related protein that enables the degradation of heme into free iron and carbon monoxide. Increased levels of this protein is thought to be a pathological feature of age-related neurodegenerative diseases, including Alzheimer's disease, and may affect neurogenesis in the hippocampus. Here the authors sought to determine how HO1 is involved in adult hippocampal neurogenesis. They used transgenic HO1 mice and adult neural stem cells to investigate the implications of HO1 overexpression, and they found that it reduced proliferation and induced apoptosis in the cells. They also found that HO1 overexpression inactivated the endogenous WNT beta-catenin pathway, and that reactivating the pathway in the overexpressing cells rescued the proliferation and survival of the cells. Phosphorylation of AKTS473 and GSK3-beta-S9 was also shown to be decreased in HO1 overexpressing cells. Overall, the group concluded that HO1 was regulating cell survival and proliferation by inhibiting the WNT beta-catenin pathway through repression of AKT-GSK3-beta. The next paper is called Upregulation of RAN3 Induces Endosomal Dysfunction in Alzheimer's Disease. This is by first author Shen and last author Wu, published in Translational Neurodegeneration. As some background, about 1/3 of Alzheimer's disease risk genes encode proteins related to endocytic function. One of these is RAS and RAB interactor 3, or RIN3, which is a guanine nucleotide exchange factor implicated in both early and late onset Alzheimer's disease. This group aimed to figure out exactly how RIN3 is implicated in AD pathogenesis using a combination of cell culture and mouse models. In APP-PS1 mice, they found that increased levels of RIN3 mRNA in both the hippocampus and the cortex. They also found increased RIN3 and early endosome enlargement in cholinergic neurons cultured from APP-PS1 mice. Getting more into the pathogenic mechanism, they found that RIN3 was able to recruit BIN1 and CD2AP to early endosomes, and overexpression of RIN3 in cells promoted APP cleavage and increased p-tau levels. All of these effects were rescued by expressing a dominant negative RAB5 construct in the cells, indicating that RIN3 was acting through RAD5 to impact endosomal trafficking and signaling. So moving away from the signaling pathways and into the larger scale of brain network connectivity, we have the relationship between neuropsychiatric symptoms and default mode network connectivity in Alzheimer's disease. This is by first author Lee and last author Lee, published in Psychiatry Investigation. So this group set out to investigate the relationship between neuropsychiatric symptoms and default mode functional connectivity in Alzheimer's disease. They assessed neuropsychiatric symptoms in 70 patients with AD using the neuropsychiatric inventory and conducted seed-based functional connectivity analyses using functional MRI to identify default mode networks. They found that there was a significant correlation between hyperactivity syndrome and decreased connectivity in the medial prefrontal cortex of the anterior default mode network. Hyperactivity syndrome was defined as agitation, irritability, aberrant motor behavior, euphoria, and disinhibition. This study suggests that specific network alterations may be associated with specific neuropsychiatric symptoms. The next paper is by first author Pohl and last author Furman, and it was published in Nature Neuroscience. It's titled Memory Trace Interference Impairs Recall in a Mouse Model of Alzheimer's Disease. As a bit of background, a memory engram is the term given to a group of neurons that encode a memory. This has been a hot topic since the previous discovery that artificial activation of an engram can restore memory in a mouse model of AD. Here the authors wanted to find out more about memory engrams in the context of AD. They used a 2-photon in vivo imaging technique in APP-PS1 mice that expressed GFP under the FOS promoter to visualize neuronal excitation, and they had mice perform a hippocampus-dependent memory task. During recall, they found that the CA1 engram was partially reactivated, even under AD-like conditions. In the AD-like conditions, however, they found a novelty-like ensemble in the CA1 region that compromised recall by interfering with the engram. Mimicking that novelty-like ensemble signal in the wild-type mice expressing GFP under the FOS promoter also reduced recall, and reducing the novelty-like signal in the APP-PS1 mice rescued the recall impairment. Next up is a paper on taste dysfunction in AD, titled Selective Peripheral Taste Dysfunction in APP-PS1 Mutant Transgenic Mice. It was by first author Wood and last author McPherson, published in the Journal of Alzheimer's Disease. Previous work suggests that taste dysfunction occurs in early stages of AD. This group sought to investigate this further using the APP-PS1 transgenic mouse model, and used the brief access gustometer test to evaluate taste responses. They also performed staining of the tongue, gustatory ganglion, and the brains of the mice. They found a significant reduction in bitter taste sensitivity in APP-PS1 mice compared to wild type, and showed that the mice also had a loss of TRPM5 expressing taste receptor cells. They didn't observe a loss of neuronal cell bodies within the primary gustatory sensory neurons, but suggested that degeneration of peripheral axons innervating the taste bud may influence the loss of the TRPM5 expressing taste receptor cells. Overall, the study supports a role of peripheral taste dysfunction in AD through the selective loss of taste receptor cells. The next paper did a very similar study in a different mouse model, and this study was by first author Narukawa and last author Misaka, and was titled Analysis of Taste Sensitivities in APP Knock-in Mouse Model of Alzheimer's Disease. It was also published in the Journal of Alzheimer's Disease. This study investigated the effect of cognitive impairment on taste sensitivity in 12-month-old APP knock-in mice using behavioral assays and a series of other measures. They found no significant difference in taste sensitivities between wild-type and APP knock-in mice, and there was no apparent difference in the expression of taste markers in their taste bud cells. So from here on out, it's a pretty mixed bag of topics. So starting out, we have a paper called Relationships Between Big Five Personality Factors and Alzheimer's Disease Pathology in Autosomal Dominant Alzheimer's Disease. This is by first author Aschenbrenner and last author Hassan Stab, published in Alzheimer's and Dementia. So since personality changes caused by AD can be hard to disentangle from changes associated with normal aging, this study leveraged the younger, dominantly inherited Alzheimer's network, or DIAN, cohort to investigate how changes in personality traits may be markers of Alzheimer's disease. They determined personality traits in the DIAN participants with the International Personality Item Pool at baseline, and related them to years to onset of clinical AD symptoms and AD biomarkers. They found that neuroticism and conscientiousness were correlated with both years to symptom onset and markers of tau pathology in the CSF. They also found that participants who were rated as higher on neuroticism and lower on conscientiousness had faster rates of cognitive decline. This indicates that assessing neuroticism and conscientiousness may be useful in mapping AD progression. Next is a paper called Features of Postnatal Hippocampal Development in a Rat Model of Sporadic Alzheimer's Disease. This one's by first author Rudnitskaya and last author Kolosova, published in Frontiers in Neuroscience. In this study, the authors wanted to test the hypothesis that specific features of early postnatal brain development may be prerequisites to AD later in life. To do this, they used the Oxy's rat model of late onset AD and compared them to control Wistar rats. They found that there was a shorter duration of gestation in the Oxy's rats, which was accompanied by smaller litter sizes and lower birth weights compared to control rats. They also found that the Oxy's rats had decreased locomotor activity and increased anxiety at a younger age. Upon examination of the brain, they found delayed neuronal maturation in the Oxy's rats, which led to changes in the mossy fiber formation. Altered astrocytic migration was also observed. The group postulated that these early alterations may be connected to the development of the AD-like pathology in the Oxy's rats later in life. Next up, we have a paper called SRC1 Knockout Exerts No Effect on Amyloid Beta Deposition in APP-PS1 Mice. This is by first author Wu and last author Li, published in Frontiers in Aging Neuroscience. So steroid receptor coactivator 1, or SRC1, is related to major AD risk factors like a decline in estrogen and aging, and it has been found to be abundant in the hippocampus and implicated in cognition. So this group set out to determine whether SRC1 is directly involved in the AD pathogenesis. To do this, they crossed SRC1 knockout mice with APP-PS1 mice and looked at the expression of synaptic proteins, A-beta deposition, and the activation of astrocytes and microglia in the hippocampus. They found that SRC1 knockout didn't have a significant effect on any of their outcome measures. The authors noted, however, that SRC1 may have an effect on tau hyperphosphorylation, which was not investigated here, and that there are considerable limitations to their study design given the mouse model used. The next paper was a pretty foreign concept to me, and it was titled Basophilic Invasion in the Neurohypophysis is Increased in Patients with Alzheimer's Disease. This is by first author Johnson and last author Biltman, published in Clinical Neuropathology. So this group aimed to determine whether there is migration of cells known as basophilic invasion in the posterior lobe of the pituitary gland, which is also known as the neurohypophysis, of patients with neurodegenerative diseases. They evaluated 122 pituitary glands post-mortem, including 45 from patients with Alzheimer's disease. They observed basophilic invasion in 78% of the Alzheimer's disease cases, which was significantly higher than the 51% of the age-matched controls in which they observed this. They did not find that basophilic invasion was associated with nestin activity, and they proposed that evaluating basophilic invasion as a marker of Alzheimer's disease may be useful, and that it warrants further investigation. And finally, last for this episode is a paper called The L1 Cell Adhesion Molecule Affects Protein Kinase D1 Activity in the Cerebral Cortex in a Mouse Model of Alzheimer's Disease. This is by first author Chen and last author Zhao, published in Brain Research Bulletin. So previous work from this group showed that the cell adhesion molecule L1 and phosphorylated protein kinase D1, or PKD1, are co-localized in cultured neurons, and it seems that these two proteins are neuroprotective under pathological conditions. In this study, they investigated the functional relationship between L1 and phosphorylated PKD1 using a combination of techniques. Starting with the human brain, they did a cortical tissue microarray with the frontal lobe of an AD brain and compared it to control human brain tissue. This showed increased levels of both proteins in the AD brain, which correlated with each other. They also found converging results in the APP-SWE mouse model. Then they used cultured cortical neurons to show that adding recombinant L1 increased PKD1 phosphorylation and maintained neuronal survival after exposure to oligomeric A-beta-42, even when PKD1 activity was inhibited. This suggests that L1 may play multiple roles in promoting neuronal survival. And that concludes this episode. Thank you so much for tuning in, and I hope you enjoyed it.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind Aminder. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the Aminder team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, yeah, your feedback matters to us, so please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.